this video, we would like to talk about color blindness. Now, 1 in 12 men and 1 in 200 women are affected by some form of CVD, or color vision deficiency. This affects about 4.5% of the entire world population. There are multiple potential causes of color blindness, the most common being a genetic disorder, where the condition is inherited from the mother of the individual. In this case, the genes that cause color blindness are the X chromosomes. Because color blindness is a recessive trait, it can be overridden by another dominant X chromosome. For example, females have 2 X chromosomes. If the recessive trait is present in one X chromosome, the other functional X chromosome is normally enough to compensate for the loss of the other. In the instance that males receive this trait, they only have 1 X chromosome and are not able to have it be overridden. This is why men are much more susceptible to color blindness than women. Other causes of color blindness include aging, eye problems such as glaucoma, macular degeneration, cataracts, diabetic retinopathy, injury to the eye, and side effects from certain medicines. Colorblind people can have many difficulties in the simple tasks of life such as driving, selecting clothing, or even choosing and preparing food. Because of this defect, these individuals may not be allowed to have certain jobs that require them to have color perception. Now that we have given some of the background on color blindness, we would like to explain how color blindness occurs. In order to properly talk about color blindness, let's look at how the normal eye functions. When light energy hits the pupil of the eye, the energy is focused onto the retina. Within the retina are special photoreceptor cells called rods and cones. Rods allow people to see shapes and shadows and assist in dimly lit areas. Cones allow people to see colors and brightly lit areas. We are going to focus on the cones as these are what allow us to see or not see color. There are 3 types of cones in the retina: L cones, M cones, and S cones. L cones allow us to see longer light wavelengths, which result as reds. M cones allow us to see medium-length light wavelengths, which result as greens. S cones allow us to see shorter light wavelengths, which result as blues. The normal eye is able to see all of the colors in the visual spectrum by utilizing all 3 of these types of cones in combination with each other. This is known as trichromacy. When light hits a cone, the energy reacts with a special protein within the cell, changing its shape. This reaction allows the protein to be turned into an action potential, or an electric impulse, that can be sent to the brain and be interpreted as what we call color. Here is an example of how the normal eye, with all 3 types of functioning cones, sees color. In the instance where someone has an abnormal or absent cone type, they will have a form of color deficiency. This is known as anomalous trichromacy. When the red cone photopigment is abnormal, the individual is suffering from protonomy. Reds, yellows, and oranges look green, and colors are dulled. When there are no working red cone cells in the retina, the individual is suffering from protonopia. Reds are perceived as blacks, and different shades of orange, green, and yellow all look yellow. When the green cone photopigment is abnormal, the individual is suffering from deuteranomaly. Greens and yellows seem red, and blues and violets are indistinguishable from each other. This is the most common form of color blindness, known as red-green color blindness. When green cone cells are not working, or are absent in the retina, the individual suffers from deuteranopia. Yellow and greens are more of a duller beige, and reds are brown. When the blue cone photopigment is abnormal, the individual suffers from TRITONOPIA, an extremely rare form of color blindness. Yellows and reds are hard to distinguish from pink, and blue looks greener. When the blue cone cells are not working or are absent in the retina, the individual suffers from TRITONOPIA. Yellow seems violet or gray, and blue, once again, looks green. This is known as blue-yellow color blindness, and is also extremely rare for an individual to receive. In very rare cases, an individual can have what is called monochromacy, where the afflicted do not see color at all. The 2 types of monochromacy are cone monochromacy and rod monochromacy, or ACHROMOTOPSIA. When afflicted by cone monochromacy, 2 of the 3 cone cells do not function properly. The individual has difficulties in distinguishing colors because the brain requires comparisons between signals of different types of cones to see color. When only one type of cone functions properly, the brain cannot make the comparisons. There can be red, green, and blue cone monochromacy. When afflicted by rod monochromacy, no cone cells have working photopigments. This means the individual sees only blacks, grays, and whites. This is the most severe type of color blindness, and is also very rare. While color blindness has been considered incurable, one of the most notable attempts was done by J. and Maureen Neitz, who had taken 2 male squirrel monkeys, which are born without the ability to tell reds from greens, and injected a human gene that detects red color behind their retinas. After the procedure, the monkeys were able to distinguish between reds and greens, getting their tests right most of the time. While the procedure is not quite ready for humans yet, it is quite a step in the right direction. There are also certain eyewear people with anomalous trichromacy can use, such as the achromo glasses. The lenses in this eyewear saturate the color the user sees, allowing the normally dull or indistinguishable colors to differentiate themselves from each other. Color blindness is a very interesting yet troublesome phenomenon. Hopefully sometime in the future, we will be able to create a technology or procedure that can fully cure or at least help those with color deficiency. Thank you for watching.